Good afternoon, everyone. My name is Tamer, Mohamed, CEO of Aspect Biosystems. Our company is based in Vancouver, Canada, and we're focused on developing what we call bioprinted tissue therapeutics. We really see this as a new category, the next generation of regenerative medicine. So just starting right at the top in terms of the mission of the company, what we're focused on doing at Aspect is bioengineering allogeneic tissue therapeutics. So these are cell-based implantable tissues that we surgically insert into the body to replace, repair, supplement biological functions inside of the body. And so to create these bioengineered tissue therapeutics, we combine our proprietary computational tissue design, our microfluidic bioprinting technology, our fit-for-purpose biomaterials, and of course, at the very heart of our tissue therapeutics, the therapeutic cells themselves that are delivering the therapeutic effect. Just as a brief overview of the company, Aspect is based in Vancouver. We have over 80 brilliant scientists, engineers, therapeutic developers, and entrepreneurs at our company. We have built and continue to grow a very strong IP portfolio with over 80 filings to date. We have our own internal pipeline focused on metabolic indications, and of course, we also focus on external partnerships, focus on bringing different therapeutic tissues to clinical evaluation and ultimately commercialization. So just as a brief overview to our platform technology, what we've created is a full-stack tissue therapeutic platform. And what I mean by that is we've integrated all of the functionality that we need to go all the way from the design of these tissues in a computational setting to the manufacturing to the implantation. So the entire process really starts in a computational setting where we're able to really design these tissues in software. We're able to really specify the overall structuring, the architecture, the materials, the properties that influence the overall functionality. Then we move into the second vertical, into the real world, where we essentially take cells, the therapeutic essentially component of our tissues, and we combine those with different biomaterials. So we have multiple different materials that we incorporate in our tissues that provide different functions. For example, immune protection or encouraging vascularization in the body or providing materials that the cells are going to live in and be happy. And so we take all of these different cellular and biomaterial inputs and then we bring them into our microfluidic printing technology. And so that's where we're able to really combine, mix, sequence through computer control, all of these different materials. And in step 3, we're able to bring that cell-loaded biomaterial out of the printhead in a very rapid fashion and essentially build up a 3D structure according to the software design that we specified in step 1. And so we take this cell-based tissue structure and then we implant it into appropriate surgical sites into the body, and it ports in and ultimately engrafts to provide essentially a functional cure for the specific disease indication that we're focused on. So just to give a brief video of the printing process that's a part of our tissue therapeutic platform, what you could see here is multiple different colored input materials that are brought into our printing technology. So these would represent the different input materials, including the materials that have the cells. They're all brought into liquid form, and then we bring in this yellow cross-linker that solidifies that material into a solid cell-loaded fiber. And we need a solid so we could build a 3D structure. And so what you could see here is we can very quickly dispense these materials. So this cell-loaded fiber is able to be patterned into a 3D structure according to the design. And so what comes out of the printer is the living cellular material. And so we're creating these combination products in a single step. And in a matter of minutes, we could produce a functional tissue therapeutic. So developing a combination product, a complex cell therapy that has to biologically function inside of the body, be immunoprotective, engraft, be surgically implanted, be retrieved if and when needed. It's really a multivariable problem. So to solve the multivariable problem, we've created a multi-parameter optimization system so that we could optimize all of these parameters and create a system that could address that multivariable problem. And so what you see on the left-hand side is a visual of our tissue therapeutic at the macro level. We could specify all of the properties that influence the functionality. At the micro level, we are able to specify the structure, the composition. You could see a representative micro sort of fiber at the top of that left-hand side, showing the cells in the core surrounded by several other materials that have different purposes. So in the core, we're able to load the cells in a material that those cells really like and could function. So here's an example where you could see islets that are functioning as demonstrated by dysosome staining or insulin positivity. Then we introduce other materials around that. For example, shell 1 is an immunoprotective shell, and when we show a cross-section of that tissue, looking at the fiber, you could see that we don't have any immune cell infiltration, but we could also get insulin out and glucose in. And then finally, shell 2 is a completely different material that is built essentially to encourage engraftment inside of the body and optimize the host tissue response. And so really what we're able to do is, again, design these tissues in a computational setting, rapidly manufacture these designs into high-fidelity bioprinted tissues where the cells are viable, printed at appropriate densities, and are functional, and then ultimately implanted into appropriate surgical sites. And so these are combination products that are, of course, very, very complex. And so we've had to develop a toolbox of sophisticated technologies to help us manufacture these at extremely high quality. So this is an example where we've essentially developed our own proprietary quality by machine vision technology that allows us essentially to monitor the cells as they're coming down from the print head. And then throughout that tissue structure, we can know if there are any issues that are occurring and essentially have a almost full, complete inspection of our tissues using artificial intelligence. So switching gears to the application of the platform, we've been really focused on applying our therapeutic platform across multiple different organs and diseases. The two main organs that we're focused on are the pancreas and the liver. Pancreas focused specifically on type 1 initially, but also we see opportunities in type 2 and obesity. And in the liver tissue space, we're developing a platform, liver tissue therapeutic that we could apply to both acquired and congenital disease indications. And we have several other areas at the early discovery stages as well. And so for the past period of time, we've been really focused on applying the technology to these different indications and demonstrating therapeutic effect and efficacy. And so we've been successful at showing early promising data that is giving us strong reasons to believe that essentially we could advance these different programs to later stages of development and ultimately clinical evaluation. So the first indication that we're focused on, and one of our more mature programs, is focused on type 1 diabetes. So type 1 diabetes, as I'm sure everybody here is aware, is of course a very serious disease that affects millions of people around the world. And these people who live with type 1 diabetes have lost the precious islet cells in their body that are responsible for sensing glucose and releasing insulin. And so they have to rely on daily insulin injections and other technologies to maintain their blood glucose. And so really our goal here is to provide a functional cure, a bioprinted tissue therapeutic that we could implant into the body that could essentially self-regulate blood glucose levels without the need for immune suppression. And so here we're showing efficacy in an immune-competent rodent model. And so maybe I could quickly take us through this data. And so on the x-axis is time measured in days post-implantation of our tissues, and on the y-axis is random blood glucose. And so we use the standard STZ model here to induce disease. So that injection results in a high rise in blood glucose. And on day 0, we implant our bioprinted pancreatic tissues into the omentum of these animals. And we could see that very quickly we could bring the blood glucose levels down to normal levels. And on day 30 and day 60, as well as day 90, we remove the tissues and we demonstrate that removal of the tissues leads to hyperglycemia or a return to disease state, showing unequivocally that our tissues are responsible for efficacy. But it also shows that we could successfully remove these tissues from healthy animals, which is an important safety consideration as we incorporate stem cells in these therapeutics. And we've, of course, demonstrated this bioprinted pancreatic tissues and many other disease models or many other animal models of type 1 diabetes. And this is just an example of how we've demonstrated efficacy across multiple different animal models. These are rodent models that I'm showing here, both immune deficient, immune competent, also looking at a xenogenic context as well as an allogeneic context. And today, actually, we're announcing an exciting deal. This was just announced a few minutes ago. The press release went live. We're incredibly excited to partner with Novo Nordisk on this huge opportunity. We couldn't have chosen a better partner. And so working with Novo Nordisk, we'll be developing bioprinted tissue therapeutics for diabetes and obesity. The initial focus will be on developing bioprinted pancreatic tissues for type 1 diabetes. The partnership will focus on really leveraging our core capabilities in the tissue therapeutic space, leveraging our bioprinting technology, and of course combining that with Novo Nordisk's significant expertise in cell therapy and therapeutic development and stem cell manufacturing, and ultimately bringing this, we hope, tissue therapeutic to clinical evaluation and ultimately the patients that are, of course, inspiring us to do all of this hard work. And so again, we're incredibly excited about this, and from an economics perspective, the partnership involves a US $75 million in upfront and $650 million US in future milestone payments per product, and the partnership contemplates up to 4 different products in the diabetes and/or obesity space, which brings us to an overall deal value of north of $2.6 billion plus tiered royalties. And so I definitely encourage you to read the press release, and we're very, of course, excited about this and looking forward to working side by side with Novo to bring curative therapies to patients that need them. So shifting gears to liver. So liver is a proprietary program of ours. We're developing liver tissue as a platform that we could apply to multiple different indications in the liver space. And the way we look at our liver indication strategy is breaking it down into 2 large categories. The first is liver failure, which splits out into acute on chronic and acute liver failure, as well as the second category, monogenic liver disorders and diseases, where we're looking at a host of different indications, all of which are rare diseases, and we aim to take advantage of accelerated pathways into the clinic via orphan disease designations. And so the first indication that we're focusing on is acute liver failure. And so this data here demonstrates efficacy in immune-competent mice models of acute liver failure. So this is using a carbon tetrachloride-induced liver failure model. On the left-hand side, you could see this data. On the x-axis is days post-surgical implantation of our bioprinted tissues. On the y-axis is probability of survival. And these tissues on the left-hand side are implanted into the interperitoneal space, and we could see a 67% increase in survival when we compare the bioprinted liver tissue arm via the cell-free control. We're also looking at different surgical sites, particularly for indications like acute liver failure, where the patients are really, really sick, and we don't want to expose them to a elaborate surgery. And so we're looking at the subcutaneous location as well. And so here we show that we are able to get almost equivalent data in terms of survival in a subcutaneous context. And so here we see about 70% increase in survival in that surgical site. And as I mentioned, we're really looking at liver tissue as a platform in and of itself, where we could target multiple different disease indications that correspond to a loss of a particular function. And so here you could see multiple different clinically relevant functions that our tissues exhibit, from detoxification functions to metabolic functions, synthetic functions. And so we look forward to keeping you up to speed with readouts from the different disease animal models that we are currently running using our tissues. Last but absolutely not least, I'd like to highlight our amazing team. It's easy for me to stand up here and share all the exciting updates, but they are the minds and hearts and souls behind our phenomenal work that we're conducting at Aspect. We have both a visionary team, but also a very experienced team, people who have brought over 25 INDs and BLAs for evaluation and commercialization. And so we're extremely fortunate to work with people that, number one, enjoy working with each other and enjoy innovating and pushing the boundaries. And of course, on our board, you probably see very familiar faces. You heard from Devin just before I spoke. He's the CEO of a very exciting company, Arbor Biotechnologies, the chair of Alliance of Regenerative Medicine, and we have other great people on our board, as well as fantastic scientific advisors that are leaders in their respective areas. And so we're, again, very fortunate to have these people on our side. And so to bring it all together, Aspect is really pioneering a new category in regenerative medicine, cell therapy 2.0, bioprinted tissue therapeutics. We really feel that there's so much potential in cell therapy, and if we can marry this with advanced technology, we could pave the way for, of course, the next chapter. We've assembled a visionary team, an experienced team. Our tissue therapeutic platform is broadly applicable, but we're very laser focused initially on pancreatic and liver tissue, and the strategy is one that involves focus on partnering with global biopharmaceutical leaders, as well as developing our own wholly owned proprietary pipeline. So with that, I thank you again for listening to our story, and I do look forward to connecting with you throughout the conference. I'm here with Eric Roos as well, our Chief Business Development Officer, and Natalie Kornick, our Head of Marketing. So feel free to connect with any one of us, and we'd be happy to chat. Take care. Thanks.